Mask way down to the corner, then you don't have the red glowing top of the screen. Welcome everyone. It is my pleasure today to introduce Claudia Elkins, who is grabbing the first of her hankies already, just in case.
I want to see a wave.
Claudia is a master's student in our department, and for the last 2 years, year and a half, you've worked with me. But initially, when she came to the University of Georgia, she started out working with Susie O'Connell, who, after considering her life path, decided that she'd rather quit than not take care of her child. So Susie had to leave. Claudia came to talk to me about what to do next, and after I desperately tried to steer her towards pretty much everyone in the department, Claudia finally said the fateful words, "But how about working with you?" And I sighed, I think, probably, and said, "I'd be delighted to." And it truly has been delightful. All of you know Claudia. She has been a wonderful member of the department. She has been a great mother to many of the students in the department as well. I've already had requests to find a new lab mom. But on top of that, she has also just been able to do some really cool research, and that's what she is going to be able to talking about today. But the real reason that I am really excited to be able to introduce her today is because I finally have the opportunity, Claudia, to ask you, is it okay if I record this?
Yes, it is.
Thank you. Claudia has, I don't know how many hours of recordings of the two of us talking, so way back
and transcribed pages.
But with that, I'll turn it over to Claudia and have at it.
Thank you so much, Mark, and thank you so much for coming to my seminar today. And I especially want to give a shout out to those calling in, namely my committee member, Dr. Jennifer Bolt, who's calling in from Ohio. Yeah. Anyway, I'd like to talk about my research, where we studied LED lighting. So my overall research objective was to discover ways to provide supplemental light in the most effective and efficient way possible. So I'm really excited to talk about my research from the 3 studies I did over the past 2 years. The first study was a photoperiod study in a growth chamber. The second one was a photoperiod study in a greenhouse. And my last one was a far red study in a growth chamber. So as you may know, a lot of our research is driven by money. There was a 2017 Department of Energy report that estimates the U.S. spends $600 million per year on horticultural lighting. And to reduce those costs, we need to provide light in a way that allows for the most efficient photochemistry. When we talk about controlled environment agriculture, we're mainly talking about 2 different types of operations. Greenhouses, where a lot of growers provide supplemental light because the natural light changes over the course of the year. And in greenhouses, electricity costs can be up to 30% of the total production costs. Then we also have vertical farms or plant factories. And in this type of operation, electric light is the sole source of light provided to the plants. And in these operations, electricity costs can be 40 to 60% of total production costs, with half of that going towards lighting and the other half towards air conditioning to remove water from the building and heat from the lights. So a lot of greenhouse growers, they use this map to determine how much supplemental light, if any, they may need to provide over the course of the year. So this map shows the average daily light integral for different parts of the country throughout the year. As you suspect, in the winter, the light levels are low, and in the summer, the light levels are high. So what we want to know is if growers are using supplemental light, we want to make sure that that light is provided in the most efficient way possible. And to do that, we're really interested in studying how plants use light. And then to do that, we study photosynthesis and the processes that go on in the chloroplasts, such as photochemistry. So when we want to know where photochemistry happens, let's start with the leaf, and we'll take a cross section of the leaf, and we have the mesophyll tissue. And within the mesophyll tissue, we've got the mesophyll cell that contains a lot of chloroplasts. And it's chloroplasts that is where photochemistry happens. So drilling down further, I don't mean to laugh, but I've seen this slide, and a lot of you have seen this a lot too. So anyway, drilling down further, this is a diagram of the light reaction of photosynthesis. And light provides a photon, and with that energy, an electron from the splitting of water starts to move through the electron transport chain. And it'll stop here in photosystem 2 because that's a rate limiting step. So as long as this electron remains in photosystem 2 can't use any more light energy because the reaction center of photosystem 2 is considered to be closed. So eventually, the electron moves on through the chain to photosystem 1, and then with the help of another photon, eventually goes on to make NADPH. So the process of the movement of electrons through the electron transport chain also causes hydrogen ions to move across the thylakoid membrane, and the resulting pH gradient is used to make ATP. Okay, so this diagram shows the relationship between the light reactions in the thylakoids, where ATP and NADPH is made, and then that is used in the Calvin-Benson cycle along with CO2 to go on and make carbohydrates, which the plant then uses for growth and maintenance. So my first study, we looked at the effects of different photoperiods on electron transport through photosystem 2, and this was done in a growth chamber. So not all absorbed light energy goes towards photochemistry. Much of it is given off and converted to heat, and then a very small percentage is re-emitted in the form of chlorophyll fluorescence. And measuring chlorophyll fluorescence lets us see how well photosystem 2 uses the light energy for photochemistry. And this is one of many reasons I need to get back home to Kansas. So chlorophyll fluorescence gives us the quantum yield of photosystem 2, which is the fraction of absorbed light used for photochemistry. And then if we have that value, then to determine the electron transport rate, we need to know the photosynthetic photon flux density, or light intensity, PPFD, which is a quantitative measure of light intensity. So if we have those two things, then we can calculate the electron transport rate with the following equation, where electron transport rate equals the quantum yield of photosystem 2 times the amount of absorbed light times 0.5. And this is assuming the leaf absorbs 84% of the light that it receives. And we multiply by 0.5 because remember, it takes 2 photons to move 1 electron. Next, we coined the term daily photochemical integral, or DPI. And it is the total electron transport through photosystem 2 per day. It's expressed as moles of electrons per square meter of leaf per day. And it's analogous to daily light integral, or DLI, which is the total accumulated light per day. And DLI is expressed as moles of photons per square meter of ground area per day. So just like PPFD and ETR are instantaneous values, DPI and DLI are values over the course of the day. This graph is from a study that Jeff did on lettuce in a greenhouse. And it's a quantum yield of photosystem 2 as a function of light intensity. So generally speaking, with higher light, the quantum yield of photosystem 2 decreases. That kind of led to our hypothesis that the same DLI over longer photo periods will result in more electron transport through photosystem 2. So we sowed green towers lettuce in 15 centimeter round pots, and we grew them in a growth chamber under cool white fluorescent lights with an average PPFD of 250 micromoles per meter square per second. We controlled and monitored the environmental conditions, and we fertigated as needed. Then we used another growth chamber for our study, and it was equipped with a custom-made LED light bars with a built-in dimmable driver, a data logger that we used to control the PPFD of photoperiod, and then we measured chlorophyll fluorescence with a fluorometer every 15 minutes, and from that, then we can calculate the DPI. So this is the inside of our setup. This is the fluorometer. Here's our plant. And yes.
Mouse.
Oh, yes. Oh, gosh, yes. Mouse. There we go. Oh, yeah, those guys can't see me. Okay, sorry. Okay, so this is the fluorometer. Here's our plant. And we've got a leaf and a leaf clip. So, okay. So here's a picture of the outside of our setup. So the setup is divided into two separate sections, and we tested one plant per section per day. And once again, we controlled the environmental conditions and monitored those. And our treatments consisted of two different daily light integrals, 15 and 20 moles per meter square per day. And the DLIs were randomized between the two different sections. And each of those DLIs were tested under 6 different photoperiods. And the photoperiods were randomized between days. We repeated the study 3 times. And then we analyzed our data using linear and multiple regression in Sigma plot. So this is a diagram of our setup, where we have a quantum sensor that measures the light, and that information is sent to the data logger that then does the math that we've programmed it to do to make sure that our target PPFDs are on track. And then the data logger will send a signal to the driver, which is basically just another name for power supply, that we can control, that then controls the LED light output. So this graph right here is from one day's replicate, and it's PPFD data as a function of time for a DLI of 20. So here, this graph, it illustrates how well we can control our light. So for a 7-hour photo period, we had a PPFD of 793 micromoles per meter square per second. For a 13-hour period, we had a PPFD of 427. And for a 22-hour photoperiod, we had a ppfd of 53. So the area under the curve is the daily light integral. So each one of these photoperiods received exactly 20 moles per meter square per day of light. Okay? Now this graph is our electron transport rate as a function of time. And this is one replicate, and I picked a DLI of 20 just for illustration purposes. So in this graph, we can see that, so here the area under the curve is our daily photochemical integral, or the total amount of electrons transported through photosystem 2 in one day. Okay, so for a 7-hour photoperiod, you can see there's less DPI compared to a 22-hour photoperiod. Hopefully that illustrates well enough. Okay, so this graph is our results, and this is average quantum yield of photosystem 2 as a function of ppfd, and this is for a DLI, so with a DLI of 15. So as light intensity increased, the quantum yield decreased. And then we saw a similar trend for a DLI of 20. And then here's our regression. So basically, at higher light levels, the quantum yield of photosystem 2 decreases, which is similar to what Jeff found in his study. Next, this is a graph of our average electron transport as a function of ppfd. So, and this is for a DLI of 15. So here we get an asymptotic increase of ETR with increasing ppfd. And we saw a similar trend for our DLI of 20. But what we really want to know is how much electron transport do we get when we provide a certain amount of light. So, oh, there's our regression, sorry. So, this is our daily photochemical integral as a function of photoperiod. So here we can see that with increasing photoperiod, we get much more electron transport. And this is a DLI of 15, and then this is a DLI of 20. So the higher DLI, I mean, you get almost twice as much as with a DLI of 15. So here's the money slide. So at the 7-hour photoperiod, the DPI is about 2.5 moles per meter square per day, regardless of the DLI. But when you compare that to a 22-hour photoperiod and a DLI of 15, you get a 68% increase in electron transport, overall electron transport. But then when you compare that to a DLI of 20, we see a 120% increase. So that's a pretty big deal. So what we found is that with longer photoperiods, we got more electron transport, and that the magnitude depended on the DLI. But does this translate into more growth? Well, hopefully my next study will try to answer that question. Any questions for this study?
Looks
Laura?
like someone zooming in may have a question. We couldn't hear you, but it looked like someone was trying to say something. We may have you muted.
I have a question from Griffin, if you guys can hear me.
Yeah.
Can you go back like maybe 2 or 3 slides when you were showing the efficiency, the 60% and 200 and 100 something %?
Sure. Here, let me see. There we go. Oh, there we go. Oh.
Yeah, right there. So
Yeah.
if we look at the black dots, it seems to be that after like 16 hours, it seems that it's stabilizing is there any reason why you made a regression with a linear regression instead of a, let's say, exponential or something like that?
Well, we tried different things and this fit the best in our opinion. Oh, let me repeat the question. The question was, why did we fit a linear line to the DLI 15?
Yeah, and looking like the 20, it seems to be that, you know, after 16, things tend to maybe stabilize. Is there any reason why that there is that stabilization as well? Like, so that, let's say, is a second question. If if your study figured out that, linear regression was the better fit, so why does that happen?
So you're saying that for the DLI 15, it kind of stabilizes at around 15 or 16?
Yeah, something like that.
Well, that's a good question.
It seems the same thing that for the 16 and like 19 and 22, it seems that it reached a plateau. So I was just wondering, I don't know the answer to it.
Yeah, I don't know that I have an answer for that either, but we can chat offline later.
Thank you.
Thanks, Bruno.
Yeah.
Any other questions from anybody? Laura, I see a burning question on that face.
I was wondering why Jeff's graph looked different than yours for the quantum view of the photosystem view.
Oh, okay. Well, we had different data sets.
Repeat the question.
Oh, yeah. So Laura asked, why did my graph for quantum yield photosystem 2 as a function of PPFD look different than Jeff's? Well, we had two different data sets. He had a lot more data points than I did, and the trend was the same. So, all right. Well, I'll move on to. Oh wait, am I going down or up? Oh, up. Yeah, she cuts us like a sailor.
It's just a little slow, I guess.
Oh, okay.
Because your slides are so big.
Sorry. Am I going down? Okay.
Too far.
Do you want to go forward or
No,
back?
I'm gonna go.
Back.
Back.
Oh, okay. Yeah. Kind of forgot where I was. Here, I got it.
Okay, so up
Yeah,
and
yeah,
down.
yeah. Okay, got it. Okay. Sorry. Okay. Sorry for the technical difficulties. Anyway, so this study, we wanted to take what we learned in the growth chamber and basically transfer that out to the greenhouse for some real world application. So anyway, we had a similar objective where we looked at different photoperiods with the same DLI, but this time we were looking at a common perennial that's grown from seed, Rudbeckia fulgida. Okay, so why seedling production? Okay, 2017 Ag Census, hold on a sec. Okay, 2017 Ag Census reports that there are more than 2,000 farms engaged in seedling, liner, plug, and cutting production with 345 hectares under protection and a total sales value of $644 million. That's a lot of money. So controlling the DLI in a greenhouse can be difficult because the sunlight's always changing, you got clouds, and it's highly variable. So we have an adapted LED lighting system where we can control the power to the supplemental LED lights and then measure the PPFD or light intensity at the canopy level. So once again, we've got our a similar setup where we have a quantum sensor that measures the light level That information is fed to the data logger that then calculates how much light is needed to reach a target DLI by the end of the day. So the data logger will send that information to a driver, once again, a power supply, that can then dim our LED fixtures. So if you lose your sunlight due to clouds, then the lights will come on to reach your target DLI. So once again, Jeff did a study where he looked at the effects of photoperiod on greenhouse grown lettuce, and he found that leaf size increased with increasing photoperiod. And then he also looked at measured chlorophyll content index and saw an increase with longer photoperiods. So if plants are making bigger leaves and they have more chlorophyll, both of those things benefit the leaves to capture more light and, allow the plants to grow faster. And then he also found a pretty big increase in dry weight with increasing photoperiod. So that led to our hypothesis where if we provide the same amount of light spread out over longer photoperiods, we'll get more growth. And we'll get that because with bigger leaves and more chlorophyll, plants can intercept light better. And hopefully with that, they'll have better, more efficient photochemistry. So here's a picture of our setup in the greenhouse. It's we have five separate blocks, and each block is divided equally into 5 separate sections, and the sections are separated by aluminum sheets to block the light from the neighboring section. And then we cover the whole setup with a 75% shade cloth to mimic winter DLI conditions. And then we set up a quantum sensor in each of the 5 sections in one block that will measure the light coming in, and then that information then will go to the data logger and control the light for the other blocks. So here is the layout of our blocks, and I just want to point out that one driver will control the same photoperiod in each block. Okay, so we germinated our seeds in 72 cell trays that were cut in half. We germinated indoors under a 10-hour photoperiod, and after 10 days, we put them out in the greenhouse under our photoperiod treatments, and we had 4 different treatments, 12, 15, 18, and 21-hour photoperiods, and each one of those treatments received exactly 12 moles per meter square per day of light. And then our poor, pitiful little control plants, they received a DLI of 5. We fertigated as needed, and then we harvested the plants after 45 days. So our measurements, we looked at, we used canopy imaging to measure canopy size over time. And then we took typical measures of growth, and then we measured chlorophyll content index. We analyzed our data with ANOVAs and mixed models using Jump Pro. Now here's our setup of our, to measure leaf canopy area. So basically, our setup consists of a camera with a bandpass filter, LED lights, and LED lights mounted to the top of a grow tent. So what we do is we put our plants in the bottom of the grow tent, we expose them to blue light, the camera takes a picture of the fluorescence coming off the plants. And it's a non-destructive way to measure growth or differences over time. And then we analyze the image using ImageJ software. So this is a side-by-side comparison of a black and white and a fluorescence image of the same tray of plants as an example of how cool I think this process is. So to analyze your images in image J, you start with a fluorescence image, okay? Then, I don't know if you can see this, here, mouse. So let me get rid of okay, so you can see some algae here, and here, and here. Well, that can you want to be aware of that because, you know, algae fluoresces too. And you want to get rid of that when you're calculating your canopy size. So anyway, you have your fluorescence image, then you apply a threshold to get rid of, you know, some of that algae, but you can still see it here. Then once you apply that, you get a black and white image. And here too, we can still see the algae. But when you analyze the particles, you have the option to exclude pixels of a certain size. And then you basically get a cleaned up black and white image. And then image J basically just counts all the black pixels. Okay, and once you analyze your particles, then you get an output with your total area. Now I measured I took images every week for 7 weeks. And this is a side-by-side comparison of the control and plants under an 18-hour photo period. So anyway, so this is day 1. This is August 30th, the day we put them under the treatment. This is week 2 and week 3, and you can see by week 3 you can see the differences pretty early on. So once again, the system is a pretty good system to detect differences early on. So that's week 3, 4, 5, 6, and 7. I wasn't smart enough to make a GIF, but oh well. All you young whippersnappers could have done that. Okay, this is a graph that shows our canopy size over the course of the treatments. So day 0 is the first day I put them in the treatment. And then I harvested at 45 days. And you can see that a photo period, 18-hour photo period, has the highest canopy size. Next we have 21-hour photo period, then 15 and 12. And the poor little controls, they're just pitiful. But it's a nice color. Okay, so for this graph and all the others for this study, my graphs are set up where I've got the photo period that the plants were exposed to on the x-axis, and then all my measured variables on the y. And then the control plants are represented by the dashed line of the dashed line. So for shoot dry weight, when we went from 12 hours to 21 hours, shoot dry weight increased 33%. Okay. Then for root dry weight, we had a similar result. So with increasing photo period, we had an increase in root dry weight of 22% from when you go from 12 hours to 21 hours. And then root fraction, which is root dry weight divided by total plant dry weight, there was no significance. Photo period had no significant effect on root fraction. And we measure root fraction because we don't want to, you know, grow a plant with this big, huge canopy and a little tiny root. You know, growers want a big root ball, so that can help them determine when the plants are ready to transplant. So here's a picture of all the plants from one block, side by side, just as an illustration of the poor little pitiful control plants. So that they received on average 5 moles per meter squared per day of light, and all the other treatments received exactly a DLI of 12. Okay, so for plant height, when we went from a 12-hour photo period to a 21-hour photo period, plant height increased 12%. And then we looked at we calculated compactness, which is shoot dry weight divided by height. So here, let me go back. So for plant height, you would think that, this may suggest that the plants stretched over a longer photo period, but they didn't, because when we looked at compactness, they were more compact. So with compactness, we did see an increase, and like I said, compactness is shoot dry weight divided by height. And compactness is a very important trait for a lot of growers. Okay, so for leaf area, we saw a 15% increase from a 12-hour photo period up to about 15 to 18, and then it went back down. Okay, so for specific leaf area, which is leaf area divided by shoot dry weight, we saw a decrease. So when you went from 12-hour to 21-hour, we saw a decrease of 25%, which is the opposite of what we expected, and an interesting result. Some people say that physiologists can explain anything, but we really don't have a good explanation for this one. And then I also want to point out that the control is way up there. It's got a really high specific leaf area, which is more typical of a response that we would get, because at low light levels, the leaves get thin. Okay, so we also, for chlorophyll content index, we also saw an increase of 46% when you go from the 12 to 21 photo period. And so if there's more chlorophyll, then plants can capture more light and maybe be more efficient with their photochemistry. So for this study, we found that supplementing light with over longer photo periods results in increased growth, improved root development, and with the potential to shorten cropping cycles by at least 1 week. And the potential to increase profits because growers can get more turns per year. And also reduce capital outlay because, you know, growers could install fewer lights to provide the supplemental light that they need. Okay, I'll take any questions for this study. Yes.
Can you go back to your imaging, the
Sure.
overhead imaging? Okay, it's somewhere there. I'm really surprised at how much variation you get between individual plants. These are the same cultivar, right? Do you know what kind of cultivar it is?
Yeah, this is Rudbeckia fulgida.
I mean, is it the F1 hybrid? Is it the over-pollinated cultivar? Because, I mean, normally even in our greenhouse that is not well controlled at all, when you germinate a cultivar, not wild stuff, we work with a cultivar, it's extremely uniform, the individual plants. And so it's very strange to me under this controlled conditions that you have so much variation among seedlings.
Yeah, okay, so first of all, the question is, you know, why is there so much variation among the seedlings? So, I don't know if it's an F1 cultivar, but, you know, so I took these images every week, and, you know, when you're taking the images, you know, my images might not be as uniform, is what I'm saying, is what I'm trying to say. Does that make sense?
Yeah, I mean, so can you go back a little bit?
Back this way?
Yeah. So do you think, so when you looked at the plants later and you measured, I don't know, dry weight, whatever you measured, it seems that there would be a lot of variation among, did you do it all at once or did you measure individual plants?
Oh, no, we measured them all. Okay, so the question was, do we measure Individual plants or all at once? So yeah, we took a harvest of everything.
Did you count how many plants? Because some of them look
Oh,
like
okay.
they would be totally out, overshaded.
Sure. Okay, so Cecilia asked if we counted the plants. Yes, we did. So as you can see, okay, so here, let me go back to, okay, so this is the first time I put them under the plant, or my first images. So you can clearly see that I had 36 individual seedlings per tray, right? Well then, you know, some of them just did not thrive or survive, okay? You can see that as we go through. And then, of course, then those that didn't thrive or survive, then they're shaded by the others. So the number of plants I harvested per tray ranged from maybe 22 to 36.
Was there a significant difference at the different light regimes as to how much, because it looks like those are growing way faster, so maybe more will be shaded out.
Okay, but the reports that I showed, those were per plant.
But the growers care whether you have 22 or 36 plants.
Right.
So is there a difference about the number of plants you end up with that is sellable?
Oh, so the question was, what's the question again?
Did the photoperiod affect the survival rate?
Oh, okay,
Yeah.
so did the photoperiod affect survival rate? I don't know that I can answer that question.
We did statistics on it.
Oh, well,
If you're not showing it, I assume it's not significant.
There was no effect of photoperiod on survival.
yeah, because, yeah, we did look at leaf, yes.
But in the end, was all your plants big? I mean, I guess even if they survive.
No, they were not. Like, okay, so the question was, were all the plants big at the end of the study? They were not. Because when we were harvesting, and Shane can attest to this, so what we did is we pulled out 5 representative samples of our tray of 36. And some of them, I mean, you had these little tiny, insignificant plants that were shaded by the bigger ones. So,
Generally speaking, it's an issue with perennials. If you do work with annuals, they are really uniform.
Yeah.
Most perennials are not grown from seed, partly because of this mess. It can get worse though.
it can get worse. And I'll talk about that next. Any other questions? Yeah, Rebecca?
Okay, so I see that you're comparing all these images to your control, but your control had a DLI of 5. Why did you not use a DLI of 12 for the control?
Okay, so when we did our statistics, on the treatments.
Okay.
12, 15, 18, and 20. Okay, so the question was, what was the question again?
No, but you're doing a visual comparison of, like, see, these plants are larger, but they had more light exposure.
Yeah, this was basically just for visualization purposes.
Okay.
Okay, so when we did our statistics, we didn't include the control in our statistics. I mean, we did ANOVAs just to make sure that the control really was significantly different than the treatments. So when we did our statistics, it was just the photoperiod treatments.
Why 5 and 12? Just based on the plan?
Okay, so 12 was a record.
To the other question.
Oh, sorry. Amanda asked, why a DLI of 5 and why DLI of 12? Well, the DLI 5, that was just measured. They weren't receiving any supplemental light. That's just what they got over the course of the study. The DLI of 12, that was like a recommendation for these plants.
For
And
the
so,
for this type of flower?
yes, yeah.
Griffin, one question. You used LED lights. If so, what light spectrum did you use? Was it a combination of lights?
No, these were cool white.
Cool white. Okay.
Yeah, for the greenhouse.
I have a question also. I think that most people, home gardeners, plant Rudbeckia to enjoy the flowers, not the leaves. Was there any difference, or would you suspect there would be any difference between the time from planting or transplanting to blooming among the treatments that you use to produce those transplants?
So the question was, would there be any difference?
From the time that the various treatments had been planted under conditions that you can't control outside, is there a difference in the time and the number of blooms and the color and the size of those blooms depending on the original treatment before transplanting?
Okay, so the question was, would there be any difference in the time of bloom with these photoperiod treatments at the time of planting?
Yeah.
I think I got that right. So we didn't test that. That wasn't that was outside the scope of this study. Thank you for asking that question. No, in all seriousness, yeah, we didn't transplant them outside, so. Anything else? Shane?
I have a question. So in some of the graphs that you showed, it's kind of a follow-up to what one of the other previous questions was that, you know, you are using this linear regression.
Yes.
But in some of the graphs, it does seem to show that some of these responses do seem to even out after the 18 hours. So I know that you didn't do that type of regression here, but if you had a grower that was contemplating utilizing this sort of system, would you recommend that they just stick to the 18-hour photo period or the 21? Or
So the question is, if I had a grower, would I recommend, so if I had a grower that wanted to use this kind of system, would I recommend they stick with the 18-hour photo period or the 21? Well, anywhere between 12 and 21 would work. So depending on, you know, how much money he's got to install lights, or, I mean, you can get benefit. I mean, you can get benefit at 15.
But the question is not whether you get a benefit. What would be optimal for a grower to do?
Okay. Well,
what would you recommend?
I would recommend anything in between, anything on this line.
Is that all the same?
Well, no.
Okay.
Okay, so.
Yeah, if the grower wants a better response, then they're going to want to extend that photo period. But do you think that there's going to be any benefit to them extending it up to 21, or do you think they'd be okay just staying at 18?
So the question is, would they be okay at taking it up to 21 hours or sticking with 18? Well, yeah, I think 18 is good. So, I mean, if I was doing it, just based on my understanding of this study, 18 was pretty good. I mean, when you look at the canopy size, 18 beat out 21. Heather.
I have a question about that slide.
Okay.
So up at the top, you've got the black line, which is 18.
Yes.
Then there's the 21 and the 15, and then the blue.
Yeah.
So statistically, I can see that the black is very different from the pinkish purple one at the bottom.
Yeah.
But all those seem close together, at least on the graph. How is the black one, the 18, is it statistically different than the red, the green, and the blue?
Okay, so the question was, is 18 statistically different than the other photoperiod treatments?
Right, except for the bottom.
Yeah, the control, yeah, because okay, so first of all, we did look at oh my God, light efficiency, light use efficiency. And we ran some, there was kind of a, there was a lot of math involved. And we ran some statistics on this, and there was no significance.
So
But
did you do like an ANOVA?
But so forget about this slide, go to one of your other graphs.
Okay.
Because it's much easier. So here, how did you determine what effect there was of your photoperiod treatments?
well, okay, so we ran a mixed model where we looked at photoperiod, and then we also wanted to look at block effect, because, you know, everything's out on this big greenhouse bench, you know, different, you know, you got a cooling pad on one end, and
So why did you fit a straight line and not a quadratic curve here to kind of go back to Shane's point?
okay, so first of all, we did fit our mixed model. We had a quadratic component in it, but then the quadratic component wasn't significant for 2 of my measured variables. So then we removed that quadratic component and then ran linear again, and then those were significant.
You should move on to your last study.
Yeah. Okay, so the last study was a far red light study done in a growth chamber. And we looked at 2 common perennials grown from seed, Digitalis purpurea and Rebeckia fulgida. Seedlings intercept very little of the light that's available to them in the production space. And plants this small can benefit from bigger leaves, because bigger leaves means more light interception. Far red light, as the name implies, is on the far right end of the visible spectrum in the 700 to 800 nanometer range. There's a lot of it in sunlight, and plants are quite used to it. And far red light can affect growth, morphology, and photosynthesis. Looking at the solar radiation spectrum is a good reminder that plants have evolved under a good amount of far red light. So there's a lot of reasons to add far red light, and one of those is to increase photosynthesis. When you look at McCree's data, this is the quantum efficiency over relative quantum efficiency over the different wavelengths. So McCree looked at the individual colors, and but he didn't look at any kind of, he just looked at the individual colors and didn't look at any kind of synergistic effects. So anyway, yeah, so McCree's data shows that there's, this was all under a low light, and there's less far red at low light there. So a former lab member, Shu-Young Jen, she did a study where she looked at net photosynthetic rate as a function of light intensity, and under warm white light, there was an increase. But when she added far red light, she found a significant synergistic effect. And there's another reason to add far red light. It can affect morphology, namely a shade avoidance response. So if you have a situation like that's shown in the picture, where you have an upper canopy, that'll absorb all the light and block the light down to underneath those plants. And so the plants below are shaded, and there's a lot of far red light in shade. And to avoid the shade, some plants will grow taller, and some will make bigger leaves to capture more light. This is a good example of plants with larger leaves under far red light treatment compared to plants that receive no far red. So phytochromes are important photoreceptors that measure light and light quality. They monitor light conditions and are constantly adjusting processes within the plant. It comes in two interconvertible forms, phytochrome red, which is originally synthesized in this form. It absorbs red light with maximum peak around 660 nanometers. It's considered the inactive form. So when red light hits the plant cell during the day, phytochrome red is quickly converted to phytochrome far red, which absorbs far red light with a peak around 730 nanometers, and it's considered the biologically active form that can create responses in the plant and also affect gene expression. So phytochrome far red controls many things, including growth and the shade avoidance response. So we hypothesized that providing supplemental light would result in leaf elongation, more photosynthesis, and more growth. But how much far red is optimal? That's what we hope to find out. So here's a picture of our walk-in chamber setup. It consists of 3 shelving units, each divided into 6 sections. And in all 18 sections, we installed 2 white LED light bars. And in 15 sections, we installed 1 far red light bar. And that left 3 sections with no supplemental far red as our control. So we sowed the seeds in 72 cell trays, germinated them under white LED light with an average ppFD of 186 micromoles per meter square per second. And because of poor and non-uniform germination of the Rudbeckia, I'm not going to talk about that in my results. So our design consisted of 16 different treatments. And so we had a control with no supplemental far red. And our far red levels range from 4 to 69 micromoles per meter square per second. And all the plants had a 16-hour photoperiod. So 20 days after we sowed the seeds, we turned on the treatment. They all got a ppFD of 186.4 micromoles per meter square per second, and an average DLI of 10.7 moles per meter square per day. This graph shows the basic spectral distribution of the white LED lights only. Okay? And this is it with a low amount of far red. Remember, we had a range of far red levels. Okay? And then this is it with a high amount of far red. So we harvested the plants after 54 days, and we took typical measures of growth and morphology, and we analyzed our data with linear regression and sigma plot. So for this graph and all the others for this study, I've got the levels of far red, total far red that the plants were exposed to on the x-axis, and our measured variables on the y. So with increasing far red levels, we saw an increase in shoot dry weight of 38%. And for root dry weight, we saw a similar trend. With increasing levels of far red, we had a 20% increase in root dry weight. And then here's a picture of our roots that received no supplemental far red. Here's pictures of the roots with a mid-range level of far red, and here's some from a high-range level of far red. And you can see, hopefully you can see, the bigger root system for the plants that received the high levels of far red. So for root fraction, which again is root dry weight divided by total plant dry weight, we saw a linear decrease of 16%. Okay? And then leaf area, we saw no significant effect of levels of far red on leaf area. And for specific leaf area, once again, we saw no significant effect. I'm running out of time. Okay, so for average plant height, we saw a significant increase with increasing far red. The average plant height increased by 38%, which you would think the plants would stretch, indication that they're stretched. and I'll talk about that later. So here's a picture of the plants under the control, just for height purposes. Here's one with a mid-range level of far red. And here's a photo of plants with a high range level of far red. And you can see that they're much taller. So remember, we had an increase in height with increasing far red, which you would think that plants would stretch. But when we calculate compactness, we found no significant effect, which is an indication that the plants were bigger, but just not leggy, which is a big concern for growers. So supplementing with far red light can increase growth, improve root development, with the potential shorten cropping cycles like before, increase profits by getting more turns per year. But how much far red is optimal? Well, we can't answer that because we didn't reach a saturation point. We saw positive effects across all levels of far red on the plants. So with that, there's a lot of people to thank. My committee members, Dr. Chappell and Dr. Jennifer Bolt, who's zooming in from Ohio, and especially Mark, who was generous enough to take me on after Dr. O'Connell resigned. He's continually inspired and challenged me. And I want to thank all my lab members and greenhouse mates. Michael Martin, who helped me out a lot with my first study. Pam and Sandy, who do a really good job of keeping the greenhouses up and running. Got to thank David Burley and the UGA crew, the U Garden crew, where I cut my teeth 2 years ago, weeding squash plants in a high tunnel in August. I learned what I didn't want to do for grad school. I want to thank all my fellow grad students, all my teachers, Dr. Malati, I had 4 classes with you, Mary Jane, who supports us in so many ways, and Evelyn Dodson. If it wasn't for her taking my application to Mark saying, you need to read this, because this one's different, I wouldn't have had this wonderful journey. Okay, then I want to thank my funding partners, LAMP Project, USDA, American Floral Endowment, Fluence Bioengineering. Oh, one last thought. If you're ever in Kansas, in the words of Jim Craig, the man from Snowy River, you're welcome at my fire anytime. Thank you. Any questions for my last study?
It's your last opportunity to ask her questions. She is about to just skip the stage.
I'm leaving Friday, so. Yeah, TC.
How long is it to drive to Kansas?
Driving straight through 16 hours. But I can't do that with the U-Haul.
Any questions from people zooming in? Looks like we have someone.
Oh, here. Do I need
I don't think I'm muted anymore.
I don't think we muted from this side yet.
Okay.
Anybody?
Come on, Sean's got to have a question. I see he's zoomed in.
Anybody have a question?
Bueller. Bueller?
If not, then thank you so much, Claudia.
All right, thank you.
You made it.
Very well done.
Yeah, I saw her.
Be impressive in far red. Actually, you can see, you can't see us.
Yeah, hi, good job, Claudia.
We're waving back.
We're waving and might probably
Don't want to make a liar out of it.